Today's class, the material that we talked about today will be on the test on Thursday, but there's not a ton of new information today. It's mostly using information that you already know and applying it, which I think will be helpful for getting ready for the exam. So there's a lot to talk about because we're talking about the control of blood pressure, but we're going to be using material that you've already learned and applying it to the control of blood pressure. You'll see what I mean as we go through. So the focus today is control of blood pressure, and we're going to be talking about receptors, adrenergic receptors that control constriction of blood vessels, and adrenergic receptors that control vasodilation of other blood vessels. And a reminder of the arterial pressure equation that will come in handy today and on Thursday. So this was a slide that I showed last week. The idea is that the heart and the arteries create pressure, and then it's the arterioles that control resistance, which is a factor of blood pressure. So if we go back to the equation, pressure is a function of cardiac output and resistance. Right? Cardiac output is this product right here. So the heart and the arteries control, well, the heart controls stroke volume, the heart controls heart rate. Arteries also contribute to, because they're compliant and they elasticity contribute to pressure. But now we're focusing on resistance. And the arterioles are the most important part of vascular resistance. So we're focusing on arterioles. There's millions of them, so they don't have names. But they're very important for the control both of resistance and, as we talked about last week, where the blood goes. So here is a set of arterioles that is highly vasodilated under this condition, but is highly vasoconstricted under this condition. We're going to try to tease that mechanism apart now. Let's do a clicker. I think I brought the clickers this time, so let's go ahead and do a clicker. This is asking you to apply your knowledge. Why are the pressures in the venous circulation so much systemic? We're not talking about pulmonary. Why are the pressures in the systemic venous circulation so much lower than in the systemic arterial circulation? I'm thinking of what am I thinking of? I'm thinking of a graph that is plotting pressure as a function of different parts of the vasculature. And remember, so the pressure is oscillating between a diastolic pressure of 80 and a systolic pressure of 120, for example, in arteries. And then as it gets into smaller and smaller vessels, arterioles, we have a wave that looks like this. And then it actually starts to attenuate, the amplitude of the wave starts to get smaller and smaller. And then when we get into capillary, well, we did an extremely good job of this, but you get the idea. As we get into capillaries and veins, the pressure, there's essentially no wave, and the pressure here in veins is much smaller than the pressure here in arteries. That's what I'm getting at. Show me your answers, please. let me show you the answer, and then let's talk about it. Many of you got A as the correct answer. The pressure in the arterial circulation forces blood through the arteriolar resistance, and that's what lowers the pressure downstream. So A is a correct statement. Arteriolar resistance is what the drop in pressure is over arterioles, not over the capillaries. So B is incorrect because there is not a high capillary resistance. Even though they're microscopic, or they're nanoscopic in diameter, there's a billion of them, and so the total capillary diameter is not a high resistance bed of vasculature. It's the arterioles where the resistance is significant. So A was the easier of the two between A and B. Now let's talk about C. So I gave you the equation for compliance. So compliance is equal to the change in pressure divided by change in volume divided by the change in pressure. So let's rearrange this. The change in pressure is equal to the change in volume divided by compliance. That's a simple rearrangement of that equation. If compliance is big, as I told you it is, compliance is large in the veins. And so this is a new concept. The compliance of veins, being thin-walled and stretchy, is greater than the compliance of arteries, which are thick-walled and stretchy. But the compliance of veins is high relative to the compliance of arteries. So if the compliance of veins is high, for a given pulse of volume, there is a lower pulse of pressure. And so C is a correct statement. It's using the compliance equation, which I'm not going to ask you to memorize, but I could ask you to apply it, having given it to you like I did here in this example. What the new information is that veins are more compliant than arteries. That's new information, and that's where we're going right now. And D, if nobody chose D, thankfully, because it's completely wrong. So let's talk about arterioles. Here is a cartoon. Here's a cartoon of an arteriole, a tiny artery that has branched off of a bigger artery. And it is giving rise to a network of capillaries that is providing nutrients, oxygen and nutrients, to some tissue, let's just say muscle, and it's receiving, waste products from that muscle. The blood is flowing very slowly through these capillaries into a tiny vein called a vene. These little stripes here on the arteriole are supposed to represent circular, smooth muscle wrapping the arteriole. The arterioles are endowed with a relatively generous wrapping of smooth muscle, both circular and longitudinal, but it's the circular smooth muscle that makes the difference in controlling the diameter of the arteriole. So here's the structure of an arteriole. It's got an endothelium, it's got some elastic membrane, I'm not going to ask you to memorize these names. These names were not on your anatomy coloring book list, so this is just information. The point is there's an inner layer, and then there is a smooth muscle layer. So this thick, relatively thick layer is a layer of smooth muscle, and that's what controls the diameter of this arteriole. And there's some other connective tissue layers around it. So the arteriole changes diameter in response to the needs of the muscle, needs of whatever tissue that capillary bed is networked into. Whether it's brain or skeletal muscle or cardiac muscle or liver, kidney, whatever. There are also, notice here, this little structure here and here and here, so-called pre-capillary sphincters, rings of smooth muscle that control. You can imagine, if this constricts, then the blood going into this bit of capillary will be restricted. So there's control of the arterioles, and there's control of where the blood goes. There's control of their arteriolar diameter, and there's control of which capillary gets more blood. That's the control of where the blood goes. But if we constrict a lot of these arterioles, I'm going to put the pressure equation on the board. Mean arterial pressure approximately equal to, I think I have stroke volume first. Stroke volume times heart rate, that's cardiac output, times systemic vascular resistance. If we constrict a lot of those arterioles, we're going to change resistance, global resistance. Here's a micrograph that shows you that, so the vascular smooth muscle is stained in green here. I don't even know what the red stain is, but you can see that, the cartoon of the vascular smooth muscle is based on an experiment that shows the vascular smooth muscle, both longitudinal and circular. So now let's talk about the mechanisms by which arteriolar smooth muscle controls the diameter of arterioles. It controls where the blood goes, and it controls the blood pressure. And we're going to focus on the sympathetic nervous system. So we have norepinephrine and epinephrine, the two hormones, neurotransmitters, of the sympathetic nervous system. So we talk about this a lot in the A&P1 class, which focuses on nerves, but we need to know this for now because it controls blood pressure. So I am not going to ask you to know that norepinephrine primarily activates alpha, and epinephrine activates both. But that's not a detail that you need to worry about. Let's just focus on epinephrine. Epinephrine, released from the adrenal medulla, epinephrine is a hormone of the sympathetic nervous system, and it activates both alpha and beta adrenergic receptors, two different subtypes of adrenergic receptors. An alpha receptor causes vasoconstriction. Activation of the alpha causes vasoconstriction. An alpha activation of a beta and a subtype beta 2, just to be clear, but I'm not going to ask you to know that. Activation of beta causes vasodilation. So we can vasodilate or we can vasoconstrict, depending on which receptor is activated. So now it's confusing, and I want you to work with this, thinking about maybe working in groups of, small groups, 2 or 3. Think about in what tissues are alpha adrenergic receptors highly expressed. In what tissues does activation of the sympathetic nervous system, norepinephrine, cause vasoconstriction? Now think about, okay, now apply what you know from the very beginning of this class, or even self-phys, not the beginning of this course, or self-physiology. What type of GPCR, there are GPCRs, what type of GPCR are the alpha adrenergic receptors that would cause constriction of vascular smooth muscle, of arteriolar smooth muscle? And then tell me about the mechanism. So I'm going to go downstairs and get my cup of coffee. This slide, slide 8 and slide 9, are essentially the same questions, but slide 8 is asking about alpha, and slide 9 is asking about beta adrenergic receptors. Alpha cause vasoconstriction, mechanism. Beta cause vasodilation, mechanism. Work it out in small groups, and then let's talk about it. Would like to tell me what tissues, in what tissues do the smooth muscle of the arterioles express a large number of alpha adrenergic receptors?
Digestive system.
The what?
The digestive
The
tissue.
digestive system. What's your thinking? Tell the rest of us.
Whenever epinephrine is released, that's sympathetic, so blood's going to flow to fight or flight, so it's going to flow to muscles and stuff like that, so that
Muscles, liver,
yeah. So then the core, digestive, stomach, it's
digestive
not
is
eating, so it's not.
fight or flight. So the logic is impeccable. Fight or flight. The last thing we need to do is rest and digest. So we constrict blood vessels to the digestive tract so that the blood can go to where it's needed, muscle, liver, brain. Okay, everybody clear on that logic? What type, so now we're constricting the blood flow to the digestive tract by activating alpha adrenergic receptors in the arterioles that feed blood to the digestive tract. What type of GPC are those? Some of you may have even looked it up. That was allowed. Can, it's a good, that's the second best guess.
My bad. It's constriction. So
Constriction.
it would be
GQ. Why? What's your so what was your logic?
So GQ activates calmodulin.
Yeah, it does.
It activates calmodulin.
What does it really activate?
Oh.
Let's talk about GQ. So now there's words here that we're not going to talk about, things like binding, conformational change, well, I'm talking about it, GDP, GTP, all that stuff you should be reintegrating into your brain. But what's the effector that the GTP liganded alpha Q activates?
Phospholipase C.
Phospholipase C, which
then phosphorylates.
No,
I can't remember. It cuts PIP2
into
IP3 and di.
IP3 and diacylglycerol, right? We're remembering the GQ pathway. Okay, then what happens? IP3, let's go to IP3.
IP3 diffuses into the cytosol, which
Yeah.
then activates calcium channels on the ER.
Yep. And then?
There's a release of calcium.
And then cytoplasmic calcium.
Concentration goes up, which
Yep.
then allows the tropin and tropomyosin things.
We're not in skeletal muscle, we're in smooth muscle.
The
Now the binding protein that you mentioned at the very beginning of this conversation between you and me, calmodulin, is activated.
which then goes in the dense bodies. Am I there yet? Or
You're close. What does calmodulin activate? Its abbreviation has 4 letters. Myosin light chain kinase, which phosphorylates smooth muscle myosin and activates the cross-bridge cycle of smooth muscle, which is what constriction is. So this is something to know for the test. Just heads up. So that's the type. So, okay, but we haven't finished. Well, have we finished? How do alpha adrenergic receptors cause vasoconstriction? Yeah, we've pretty much finished. We've gone from activation of the receptor to activation of the cross-bridge cycle, which is what constriction is. Dense bodies get closer together, the muscle gets shorter and fatter, that squeezes the circular muscle and constricts the blood vessel. Questions about it? This is so when I say how, I mean mechanism. And you probably have gathered by now that I'm into molecular mechanisms. So when I say give me the mechanism, I mean everything you got. Everything you got. The cell type, the receptor, the thing that activates the receptor, the G protein, the thing that the G protein, the whole gamish, okay? Just give me everything. You don't even have to write it in sentences. You can just write it in, you know, sort of bullets. You don't have to, it doesn't have to be an essay. It just has to have the information. I'm not grading you on the quality of your writing. I'm going to grade you on the quality of your knowledge and the completeness of your knowledge. Okay? So, everybody clear on this question? Okay? No? Okay. Let's talk about beta adrenergic receptor. How many of you got a chance to wrestle with this one? Because I'll give you a hint. This one's tricky. Let me give you a few more minutes to work on this. Highly expressed. Not Kendra. Anybody but Kendra. Great.
Muscles, lungs, and brain.
Muscles, vasculature, the pulmonary circulation, the heart, brain, yeah. Liver. Why liver? I mean, I know it's part of the digestive tract. So why we said alpha is high in the digestive tract, things like stomach and intestines. Why would liver be different? Like, it has to do a lot to do with glycogen. The liver is where we store glycogen. So, and glycogen is a storage molecule for glucose. And glucose is what we need for energy, bursts of energy anyway. And so we need to liberate the glucose from glycogen from liver and get it to the muscles or the brain or to the lungs where it's needed to survive. And so that's why the liver has a lot of beta adrenergic receptors. Okay, what type of GPCR are beta adrenergic receptors? We're going back to the heart here. This is not tricky. Got 1 in 3 chance of getting it right, and we're pretty much sure that it's not Q. So what do you think? GS. Beta adrenergic receptors are coupled to GS. And now your brain should be off and running on the molecular details of the activation of GS, all the way down to increase in cyclic AMP, activation of pKa, etc. Now it gets tricky. But you know the answer. Why does activation of beta adrenergic receptors in vascular smooth muscle cause vasodilation? And what I want to do is trigger a memory in your heads about, remember when we talked about the heart and beta adrenergic receptors increase the heart rate, and they increase the rate of relaxation. Because when you have an increased heart rate, you have to relax faster. Otherwise, you don't have enough time to refill between beats. So now, what was the mechanism of increased relaxation in the heart due to sympathetic activation? Because that's the answer to increased relaxation, vasodilation, of vascular smooth muscle. Anybody remember this? What do you need to relax? Somebody besides Kendra. Well, we'll go to you, Kendra, if nobody steps up. Joey? No? Want to give it a try? It has something to do with circa. Joey?
Well, I was going to say acetylcholine.
No, it has nothing to do with acetylcholine. It doesn't have to do really with the ryanodine receptor. Kendra, are you ready to give us your answer? Or have I messed it up?
I was just wondering, when you say with the vasodilation, would it have anything to link back to anything to do with actual potential skeletal muscle in the higher canal?
That's probably gonna lead you astray.
Okay.
I mean, it could. Yeah, the short answer is yes, but that's probably not the best way to get to the answer that we're looking for here. What causes relaxation of muscle? Less calcium. What causes less calcium inside a cell? Activation of SERCA, PMCA, and CX. Beta-adrenergic receptors activating PKA. PKA activates SERCA. And that's the mechanism, remember, what does PKA also do? It activates IF, or IH rather. It activates IH, and that increases heart rate. It activates calcium channels, and that increases heart rate and heart strength. It increases the closing of K channels. That increases heart rate. But it also activates SERCA, which increases the rate at which the heart relaxes and allows to reflow, and increases the relaxation of vascular smooth muscle. We're going to see this again in airway, because activation of beta-adrenergic receptors in the airway allow airway dilation. Any of you who have asthma know that you take that spray. That spray is an adrenergic activator, and it's activating the beta-adrenergic receptors in the airway, allowing airway dilation. So that's not going to be on the test. But vasodilation is going to be on the test. Questions? Your book goes into a lot of the other things that control vasodilation. So I don't want to go into this. What we've been talking about are neural and hormonal control. We've been talking about epinephrine, norepinephrine. We've been talking about, these are the two things that we've been talking about. There are other neural and hormonal controllers. I've been giving you a simplified, simplistic even, view of control of blood vessel diameter. And there are all kinds of local controllers. What do I mean by local? If I work my biceps really hard, like pretend I have a weight here and I'm working my biceps really hard, and my biceps are saying, we need more blood. And not this biceps, but this biceps. There's local mechanisms within that muscle that allows vasodilation of the blood vessels to that particular muscle. That's local. So there are local controls as well. And there's a lot known about those mechanisms, but I want you to focus on the mechanisms that we've already talked about. Epinephrine, how epinephrine is a vasoconstrictor, how epinephrine is a vasodilator. Knowing that I'm giving you the simplified view, and someday maybe you'll have to use the more complicated view, but not for this test. Okay? Clear what I'm trying to do? It's more complicated, but I want you to focus on what we have focused on. Okay, so here's a cartoon of the cardiovascular system, the heart, pulmonary circulation, circulation to the head and to the brain, circulation to the arms, circulation to the liver. You can parse this out. So on the left side of the heart, we have high pressure and lower volume. Remember at the very beginning of this, I said about 25% of the blood volume is on the left side, and about 75% of the blood volume is on the right side at any moment in time. So we talk about the left side as having high pressure. This is left, and this is right. High pressure, low volume on the left. Low pressure, high volume on the right. Where are the regions with high resistance? I think I've already given you the answer. This should be pretty straightforward. Where are the regions of high resistance? Arterioles. And where would we see arterioles on this cartoon? Well, and I'm willing to give somebody the laser to point to, and where arterioles would be on this cartoon. Yes, thank you very much.
So, for the capillaries, but
Okay, before the capillaries, but after the what?
after the arteries.
Exactly. So this this big tube represents an artery.
Okay.
These little tubes represent arterioles. So you would point, here's an arteriolar bed, here's an arterial bed. So this is a major artery. This is where the arteries are breaking down into smallest arterioles. And here, of course, is where the capillaries are. So you're right, before the capillaries, after the major arteries, somewhere in here. That's the idea. So let's talk for a minute about capillaries. Let's parse out this cartoon from your textbook. I think this one's from your text. So there's the artery, there's the arteriole. These tiger stripes are supposed to represent smooth muscle. Control of the arteriolar diameter, and the precapillary sphincters control blood flow into a capillary bed. So now we're talking about the capillary. What is important about the capillaries? What's the key thing that capillaries do? Exchange. Gas exchange, nutrient exchange. They're extremely thin walled. So here's remember in the first problem set, I asked you to calculate how fast blood was moving through the arteriolar, through an arteriole. But I didn't give you this picture, but now I'm sort of circling back. Here is an capillary. Here is a capillary. We're past the arterioles, we're into the capillaries. Here is a capillary. Here is a blood cell, a red blood cell, in the capillary. So you can see the capillary diameter is about the same as the diameter of a cell, right? This is tiny. And the cells, the blood is moving very slowly, allowing for gas and nutrient exchange. The wall of the capillary is extremely thin, so that nutrients can get into the muscle, in this case, and waste products can get out of the muscle. Gas exchange, oxygen, carbon dioxide, nutrient exchange, glucose, fatty acids, whatever the muscle needs, and the byproducts that the muscle that we need to excrete. This will not be on the exam. This is just for your information, knowing that some capillaries are tight and some capillaries are leaky. Fenestrated. Those of you who know French might know the French word for window is something like, I forget how to pronounce it, but it's, anybody know French? Fenêtre, or something like that, is the French word for window. So fenêtre, or the Latin, what am I doing? I'm just saying that it's leaky, that there are leaky capillaries. So, and you did in lab last week, we talked about how the capillaries in the lungs, remember that in the heart failure case, the lungs were filling up with fluid because pulmonary capillaries are leaky. Okay? This is a detail that we're going to need to understand when we get to the respiratory system and when we get to the kidneys. We're going to be seeing leaky capillaries. Some of them are leaky, and when I say leaky, we're not talking about leaky to nutrients or gas. They're all leaky to nutrients and gas. We're talking about capillaries that are leaky to water. Some are leaky to water and some are not. They're all leaky. They all allow gas exchange and nutrient exchange. We'll talk about nutrient exchange when we get to the digestive tract. We'll talk about gas exchange when we get to the respiratory system. Yeah?
So for penetrating capillaries, are there more pores that allow?
Yeah, there are. So in tight capillaries, there's a tight junction between the endothelial cells that make, so this is tight, and this is tight, and this is tight, and the capillary is made up of a tight endothelium. Here, there are actually holes in the capillary that allow water to pass through. Okay, so we've moved from arterioles that are important for control of where the blood goes and blood pressure. We've talked about capillaries, some capillary mechanisms that will come up on again in respiratory and kidney physiology. Now we're moving into veins. So we're just making a sort of a quick tour of the vascular system. Remember I said that arteries have a thick wall of smooth muscle. Veins have smooth muscle too, but it's not as thick. Veins are thinner, but they still have the same sort of things. They have an endothelium and they have a smooth muscle layer. Veins also have these flaps. They call them valves, but they're not as well developed as the cardiac, the atrioventricular valves or the semilunar valves. But there are flaps in the veins that help prevent backflow. So you may know, those of you who have gone on a trip to like New Zealand or Europe, if you've read the safety card, in the airplane seat, they talk about preventing deep vein thrombosis. And they tell you to, get up and walk around periodically or exercise, even just sitting, exercise your legs by extending and flexing your ankles or your knees, or do whatever you can to move your legs a little bit during a long sitting position. And that's because the muscles of the body, so this is supposed to be a skeletal muscle, like a leg muscle, help pump the blood by squeezing the blood. But because of the structure of these flaps, the blood does not go backwards, it goes forwards. So your skeletal muscle, in certain circumstances, acts like an assist to help the blood get through the veins. People who are bedridden or who are chair-ridden, who have to sit in a chair because of spinal cord injury, if they're paraplegic or quadriplegic, they have to be careful about the blood not going in the veins and generating what's called deep vein thrombosis. Thrombosis is a clot. Okay, this won't be on the test either, but you should know that muscles help the vein part of the circulatory system with blood return to the heart.
So do veins have less smooth muscle surrounding them than arteries? Like,
Veins do have less, a thinner layer of smooth muscle. They're stretchier, so they are more compliant. Circling back to this quick, veins are more compliant because they are stretchy. They have a higher compliance because they are stretchier and they have less smooth muscle. Let's take a break. And I think the rest of this class is just a series of, let's see. I've got one more slide. So there's going to be some clickers, then there's going to be a few more points, and then there's going to be a boatload of clickers that we can use to talk about. It's not a test. The test is not today. The test is on Thursday. But the clickers are used to, have an opportunity to clarify your knowledge, to see what you understand and don't understand, and ask questions. Okay, so let's take a 5-minute break, and we'll finish this up with a whole lot of quizzes. Why is the pressure in arterial circulation higher than in the venous circulation? I'll give you a hint. There's one best answer. one trick answer. I think there's a text answer. Let's see what it is. Yeah. Arteriolar resistance. Volume has little to, arterial volume has little to do with pressure. Compliance, well, that's a wrong statement. And D is a true statement, but it's got nothing to do with the difference between arteries and veins. Okay, so D is the trick. It's a true statement, but it doesn't answer the question. Don't let me trick you like that on the exam. Okay, so that was just a taste of the clickers to come. Let's try to put this together into control of blood pressure. So, obviously if we're going to control blood pressure, there has to be a way of sensing blood pressure. Your body has to be able to detect, is my blood pressure too high, is my blood pressure too low, and control it. And so this is the slide that talks about the anatomy of the detectors. A barometer is measuring air pressure. So baro is the prefix for pressure. So baroreceptors are the receptors that detect, in this case, blood pressure. And they're located in the aorta and at the bifurcation of the external and internal carotid arteries. So I'm using some of the anatomy that you know. It's on your list. Aorta, common carotid, external carotid, internal carotid. Pressure receptors are in the aorta and in the split between the internal and external carotid arteries. And now we're getting into neuroscience, and now we're getting into the other course, so we're not going to get into it here. But the point is the baroreceptors, right there, detect blood pressure. And higher blood pressure makes for higher signaling from those receptors to the brain, and lower blood pressure, this is the illustration here, lower blood pressure because of, let's just say bleeding, catastrophic bleeding, lower blood pressure, lower firing of baroreceptors. So here's a flow chart that kind of summarizes the past 3 weeks without going into the molecular mechanisms that I do want you to know. And so let's go through it. So this is in the setting of low blood pressure. But you could draw a flow chart here for high blood pressure. Let's go through it in the setting of low blood pressure. What the body needs to maintain blood. So you're bleeding from the femoral artery. Hopefully not, but somebody is bleeding from the femoral artery. Major hemorrhage, major loss of blood volume, major loss of blood pressure as a result of blood volume loss. You still want to survive, so your body maintains blood pressure in order to keep the blood going to your brain and to your heart and to the rest of your body that isn't bleeding. So how does it do that? So baroreceptors signal to the brain less, and the brain detects that and causes an increase in sympathetic nervous system activation. Bleeding triggers a fight or flight response. This is now a fight against the bleeding. Okay, so you should be able to understand how sympathetic nervous system increases the heart rate. What are we thinking? What am I thinking when I say you should be able to explain at the cellular and molecular level? Okay, what cells? Oh, it's right there, sinoatrial node. But you should be able to, even without this flow chart, the sympathetic nervous system increases the heart rate by stimulating the SA node. What receptors? Beta, is that what you said? Beta receptors. How does it increase heart rate? I think that's where you're going now. How does activation of sympathetic receptors by epinephrine and norepinephrine increase heart rate? IH, calcium channels, increased closing of potassium channels. That's, circa doesn't play a role in rate. Circa is going to play a role in force and in relaxation. But rate is controlled by IH, calcium channels, and closing of potassium channels. Okay, so I'm asking you to see what is this mechanism by which the sympathetic nervous system increases the heart rate. And here's another big arrow, increases the stroke volume, increases the force of contraction so that the volume of each pump, each beat, is increased. How does the sympathetic nervous system increase the force of contraction? Calcium. I think that's what you said. Calcium, increased voltage-gated calcium channels, increased calcium-induced calcium release, increased cytoplasmic calcium. Now we're going to get to, somebody mentioned troponin, I think that was Kendra, mentioned troponin, and because we're in the heart muscle now, not in smooth muscle, that is the correct way to think about it. Increased binding to troponin, increased allowing of the myosin cross bridges to form with actin to bring the sarcomeres of the heart muscle together to increase the rate and the extent of shortening of each myocyte, increasing the force of contraction and increasing the volume pumped out, the stroke volume. Okay, so that was all review. I didn't review all the gory details, but let me just remind you that when I ask you for mechanisms, I am asking for all the details. Just give me what you got. Don't even think twice about, do you want this or not that? It's just a few words in some cases. Okay. Increased sympathetic discharge to arterioles. Arteriolar constriction. Especially, especially the arterioles that express beta-adrenergic receptors, right? So we're going to constrict to the digestive tract. We're going to constrict to the kidney in order to conserve blood flow to go to the brain and to the muscle that might be necessary to survive this catastrophic event. We just talked about this. So this arrow here is what we talked about earlier in this class. Because increasing constriction, increasing resistance. So going back to the blood pressure equation, we've increased this, and now we're increasing this in order to maintain blood pressure. Now it gets, this one is tricky. Sympathetic discharge to veins. Constriction of veins. Why is that going to help? Let's parse this one out. Constriction of veins. Remember, the veins are compliant, and they've got a lot of the blood volume. We don't want that blood volume to be sitting in the veins. We need it now to be getting into our arterial circulation in order to not die because of the loss of blood. So your body constricts the veins, makes them less compliant, forces the blood to not be in the venous side of the circulation, increases the return to the heart, increases the blood return from the veins to the heart. That increases the end diastolic volume, right? The return of the blood from the veins increases the end diastolic volume. And now what am I thinking of? I'm thinking of the graph that plots stroke volume as a function of the end diastolic volume, left ventricular in particular. What was the shape of that curve? Did something like that. We're trying to increase the end diastolic volume in order to increase the stroke volume. Working again here. So increasing cardiac contractility and increasing cardiac refilling by this mechanism. And the veins are what control refilling. So I've explained this arrow. So increasing volume, increasing stroke volume, increasing cardiac output. So you should be able to parse this out with the molecular mechanisms that we've talked about in this class. Okay? I don't think I gave you this slide because it printed out too small to be seen anyway, but it'll be available to you on Moodle. And you should be able to walk through this with all the molecular and cellular mechanisms. So here, the target is the sinoatrial node. Here, the target is 8 arterioles. Here, the target is the heart muscle itself. And here, the target is the smooth muscle in veins. All right, I said something wrong. Here, the target, it's not just the arterioles. The target is the smooth muscle in arterioles, and you should be able to tell me the mechanism by which activation of the alpha adrenergic receptors in smooth muscle causes vasoconstriction. We did that earlier today. Okay, so I'm trying to make it clear how to best prepare for my test. Review the anatomy, use the coloring book assignments to reinforce your knowledge of anatomy. I could ask you an anatomy question that was very much like what was on the quiz, or I could ask you a different type of anatomy question. Physiology, go through these mechanisms. Be prepared to tell me, sort of a bulleted, listing way, mechanisms like we've been going through today. Pedro?
So on the problem set, the last question, I think the last question
The
was
one
drawing.
to sketch the vasculature?
So I was wondering how deep of how much knowledge do you need in regards to that? Like, for example, would you?
I would like you to know where the popliteal artery is, and if it's in the coloring book and it's on the list, you should know where it is. Be able to identify it. Same with the veins. I haven't quizzed you at all on veins yet, but it's on the list, so don't assume that I won't test you on it on an exam. It's in the coloring book. There are coloring book assignments for the veins. It's on the list. Not all the veins are listed, so you only have 8 to sort of know where they are. In fact, they're all in the arm. I'm not asking for any veins in the legs. Oh, except for great cardiac, and you know where that is. So it requires work, but that's part of your job. Okay, so here is the last slide, and that's just a reminder of what we've been talking about all class. The heart rate depends on the sympathetic nervous system that increases it, and the parasympathetic nervous system that decreases it. I'm just unpacking some of these abbreviations. Sympathetic nervous system, parasympathetic nervous system. Control heart rate. We haven't talked about how acetylcholine and the activation of GI-coupled receptors decrease the heart rate, but you should be able to tell me that happened. Stroke volume. We've talked about the sympathetic nervous system, and we've talked about refilling. Resistance. We've talked about vasoconstriction or vasodilation, and we haven't really, so you can kind of cross this out. I'm not going to ask you to tell me about local vasocontrollers. In the past, I have in fact gone through a few of those, but we're just focusing this time. I've told you that they exist, but I'm not going to test you on it. The sympathetic nervous system can cause either vasoconstriction or vasodilation depending on which of the 2 flavors of receptor are activated. We've gone through that. So let's do some clickers to try to review. We'll spend about 10 minutes with clickers. Again, this is not a test. I am not keeping track of who knows the answer. I guarantee that 2 seconds after we go to the next one, I don't remember who got the right answer and who doesn't. So this is not assessment. This is getting the conversation going. before I reveal the answers, are there questions about how I am wording this? Because that's part of your challenge during the exam, is to understand how I write questions like this. Are there questions about, when you say beginning of ventricular systole, what do you mean? Well, I mean the beginning of isovolumetric contraction. Okay, which are true? E and D. So, isovolumetric. There can be no valves open, so we can cross A out from the very start. Pressure in the left ventricle is increasing because it is contracting, but the valve, the outflow valves, the aortic, in this case we're talking about ventricular, so the aortic and the pulmonary semilunar valves have not yet opened. So pressure is increasing, but volume is not changing. That's the definition of isovolumetric. Pressure in the left ventricle is greater than pressure in the aorta. No, if that were true, that would open the aortic valve and we would not be isovolumetric. Pressure in the aorta is greater than pressure in the vena cava. Oh, vena cava.
That's just.
Well, it's true, but that doesn't, that is a true statement. The pressure in the aorta is always greater than the pressure in the vena cava. So that's a true statement. So I didn't ask you which, so if I had said which of the following explains isovolumetric contraction, D would not be true. It's simply asking you which of these are true during. Not asking you a mechanism, it's asking you at this point in time which are true, and that's why D is a true statement. You know that the pressure in the ventricle is always greater than the pressure in the veins. Especially during contraction. Okay. Next one. During ventricular ejection, which of the following are true? Okay, I'm seeing a lot of the correct answers. They're all, so I saw a lot of correct answers. I can walk through this, or I can just let you walk through it on your own time and ask me questions if you have it. Let's do that. These are all true statements. During ventricular ejection in a normal person, which of the following are true? He's got a true. Question before we reveal. Questions about the question? Yes, Mike. Did I mean QT? Yes, the EC. I'm talking about the QT segment, which is baseline. A, B, and C. Let's walk through this one. During ventricular ejection, we're after the Q wave, we're after the QRS complex, we're before relaxation, so we must be in the QT segment, between the QRS and the T wave. The heart is quiet. The only time the heart would be making a sound now is if you had a murmur. Because we're having laminar flow out of the aortic valves.
Shouldn't be ST, isn't QT also QT?
Oh, you're right, you're right. ST segment or QT interval, I've mixed them up. Both the QT interval and but it should be, the better answer here would be ST segment. So yes, the ECG signal is at baseline, ST segment. You're right, I'm sorry, that's my mistakes. The QRS is the complex, the ST segment is when the heart is at baseline between the QRS complex and the T wave. Sorry, ST segment.
Would you call it the QT segment in a lead that doesn't have the
No,
last part?
no.
expressed?
Oh, I see. So is that how I wish I could say that that's how I got confused myself, but no. You would
Okay.
still call it the ST segment even if the lead didn't have an S wave.
Okay.
Ventricular volume is decreasing because we're talking about ejection, so that one comes natural. Atrial volume, it turns out, so this was the trick, atrial volume is actually increasing because the atria are refilling while the ventricles are pumping. I wouldn't throw that kind of curveball at you, just to be clear. Okay, which of the following drugs, let's do a little pharmacology, which of the following drugs could be used to treat chronic high blood pressure? You know that there are people who are walking around whose blood pressure is abnormally high, even though they are not, in sympathetic nervous system stimulation. They're just sitting on the sofa watching TV. Their blood pressure is higher than it should be. Your grandparents, maybe even your parents, have this. Which of these drugs would be effective for treating chronic high blood pressure? Some of them are better than others. I'm not asking you to be a diagnostician here, but I'm talking about which of these mechanisms would work. This is a hard one. It's asking you to apply your knowledge but applying your knowledge to understanding mechanisms. I'm not seeing any fully correct answers yet. Are you with it? Like I said, this is hard. I'm still not seeing any fully correct answers. Now I am. Let's go through it. And I think there's some more, but we won't go through them. You can do them on your own at home because it'll be on Moodle. Blocking a beta. So these are beta blockers. You've heard of them. Drugs that block beta adrenergic receptors are beta blockers. They would block the beta adrenergic system from increasing stroke volume or increasing heart rate. Both of those would tend to decrease pressure. By blocking the sympathetic nervous system's tendency to increase blood pressure, beta blockers allow lower stroke volume, lower heart rate, lower mean arterial pressure. They also cause some vasodilation by blocking the beta, beta receptors do not cause vasodilation because we're getting into technology. The beta blockers affect these two things, and that would affect mean arterial pressure. Blocking calcium channels, lowering heart rate, lowering stroke volume, lowering vasoconstriction would all lower pressure. Is that clear? Remember, calcium channels participate in the rate, calcium channels participate in the strength, and calcium channels participate in vasoconstriction. So if we block them, we And by blocking, remember, we're not blocking them completely, that would kill you, but pharmacology is to inhibit them, to not, but when a pharmacologist like me talks about block, we don't mean 100%. That's death, right? In this case, we're talking about a pharmacological blockage. Drugs that block calcium channels in arterioles, right? I sort of lumped B and C together when I was doing calcium channels. And drugs that cause decreased blood volume by increasing urination. Remember, if I decrease blood volume, I'm going to decrease blood volume. I'm going to decrease stroke volume, and that will assist in decreasing blood pressure. So just so you know, these are all drugs that are used to treat chronic high blood pressure. Some of your grandparents are on maybe all 3 of these classes of drugs, depending on the severity of their disease. And so I'll end with one bit of information that will not be on the test. If the drug ends with olol, it's a beta blocker. If the drug ends with, well, there's many different classes of calcium channel blockers, but, well, if it ends with Zem, it could be a calcium channel blocker. And I'm talking about the generic drug name, I'm not talking about the trade. So metoprolol is a beta blocker, that's a common beta blocker. Diltiazem is a calcium channel blocker, and Pril are often drugs that increase diuresis. This, of course, this will not be on the test. If you go to your grandparents and you say, "Grandma, show me your pill, show me what pills," you'll be able to see whether they're taking beta blockers or not. Okay.